Okay, now we're going to do a unit on human genetics. Most of this will deal with pedigree analysis from chapter 6, and we'll discuss chromosomal abnormalities from chapter 4 for sex chromosomes and 8 for autosomes. So model organism for genetics research should have a short generation time, and in fruit flies that can be as little as 10 days, often 14, however. You want an organism that can produce large numbers of offspring so you can get your ratios from crosses, and a female Drosophila can lay lots of eggs. And of course, our own species doesn't fit that pattern, so we have to study human genetics in a different way. This shows some of the many advantages of using fruit flies to study genetics. So mostly what we'll discuss in this unit is pedigree analysis, and I'll show you how to read and interpret pedigrees. Also, we can use twin studies. So monozygotic are identical twins, and of course, they're genetically identical people, and anything that they differ by has to be caused by the environment. And then adoption studies are unrelated people in the same environment. So if they show some of the same things, then those had to be caused by the environment. So you can study human traits with pedigree analysis. It combines genealogy with the family history of particular traits of interest. And there are really two main goals: the pedigree analysis to determine if the trait is genetic, and then to determine it's a mode of inheritance. And I will define those modes shortly. So in a pedigree, unaffected people, that is people that don't have the genetic disorder or the phenotypic trait of interest here, are shown by usually white or open symbols. Affected people are shown by colored in symbols, not always red, but could be black or something else. People that are carriers are shown with the dot there. Males are squares, females circles, and people of unknown sex are diamonds. Asymptomatic carriers, as we can see here, have a circle in them. Multiple people, numbers in the symbol, and then deceased people have a slash through the symbol. The first affected family member coming to the attention of the geneticist is called the proband, and they're often delineated with a P and an arrow. Somebody who we don't know their family history, question mark within the symbol, and then a family. Roman numerals denote generations, and birth order is read left to right. So in generation 1 here, mom and dad are unaffected, and then they have a son followed by 2 daughters in generation 2. So the horizontal lines between the parents, of course, indicate mating, and the vertical line indicates ancestry from one generation to the next. Adoption is shown by these brackets here. At the bottom there, identical twins are shown with a little horizontal line connecting them. Non-identical twins lack that line. And if we don't know if the twins are identical or not, we put a question mark there. And then consanguinity is a sort of a fancy term for inbreeding, and 2 horizontal lines indicate consanguinity. In this case, generation 3, we've got marriage or between 2 cousins. So our modes of inheritance then include autosomal recessive, autosomal dominant, and X-linked recessive. Those are the 3 most common, and we'll limit our discussion to those. There's also X-linked dominant, but that's very rare, and Y-linked, which is also very rare. So we're not going to discuss these last 2. So first I'll list some characteristics of autosomal or of the mode of inheritance, the particular mode of inheritance, show you some pedigrees, give you some examples then of genetic disorders that follow this mode of inheritance. So an autosomal recessive, unaffected or normal parents can have affected offspring. If it's in your family, you've got about a 25% risk of inheriting the disorder. 2 affected people cannot have an unaffected child. Both sexes are affected with equal frequency because it's autosomal. There may be some consanguinity or inbreeding in the family, and it skips generations normally. So this is autosomal recessive, so we can see generation 1 there, mom and dad are normal. Then their first daughter is person number 2 in generation 2, she's normal. and their second daughter is affected, and then their son, number 4, is unaffected. And you can see in generation 3, we've got mating between first cousins, individuals 3 and 4 in generation 3 produce 2 sons and 2 daughters, and son number 2 is affected. So here's an autosomal recessive pedigree, and we notice that one individual in generation 1, that's the mother there, is affected, and then the grandson in generation 3 is also affected, and notice it skips generation 2. So one of the things I may ask you to do, in addition to telling me mode of inheritance, is to give me genotypes. So we know that since mom is affected in generation 1 there, her genotype has to be a, and we can pretty much deduce that dad is big A, because if dad was big A, they'd probably be some affected individuals in generation 2, and they're not. And so for the kid, the grandson in generation 3 to be affected, he's got to get a little a from each parent, right? So that tells us that mom and dad are each big A. So unaffected parents can have affected children. So in this pedigree, both mom and dad are affected, so both mom and dad are a, and that means all their kids are going to be a. Some examples of diseases. Albinism, that's on the long arm of chromosome 11. I don't expect you to memorize these numbers, but I just want you to have some idea of what traits are more common in what groups of people. So 1 in 37,000 people of European descent in the U.S. are albino, where it's about twice as common in African Americans, about 1 in 15,000. And then among some Native American groups, it's quite high. Among the Hopi and Navajo of the southwestern U.S., 1 in 200, so that's 0.5%. And then among the San Blas Indians in, I believe, Panama, it's almost at 1%, 1 in 132 people. Cystic fibrosis, much more deleterious. The gene's on the long arm of chromosome 7. 1 in 2,000 people of European descent have the disease, with 1 in 22 being carriers, that is heterozygotes, big A. So it's a disease of people of European descent, mostly white folks, in other words, as opposed to sickle cell, which is a disease mainly of people of African descent. It's on the short arm of chromosome 11. They say that 1 in 500 African Americans is affected by it, with 1 in 12 being carriers. And remember, carriers are selected for because they're resistant to malaria, carriers again being heterozygotes. So here's a pair of kind of famous musician albino brothers, Johnny and Edgar Winter. Johnny on the left there was a big blues guy, and Edgar Winter had some hit rock songs in the 1970s. So this shows you where the cystic fibrosis gene is, kind of the middle of the long arm of chromosome 7. Lots of different mutations can cause the disease. At the top here is the cystic fibrosis protein, CFTR, which stands for cystic fibrosis transmembrane receptor protein. It's a protein that forms a pore or a hole in the membrane of certain cells. And so the two ends of this thing would be attached together. And the numbers here are the exons in the cystic fibrosis gene, and that delta F508 tends to be one of the most common mutations causing cystic fibrosis, again, mainly found in white folks. Sickle cell is just a single mutation in one base pair that causes the change in one amino acid in the protein. So the valine down at the bottom there in yellow is a glutamate in most people. So most folks outside of Africa have 2 glutamates there, but the people with the sickle cell trait have a valine there. Now moving on to autosomal dominant. Affected parents can pass it on to affected offspring. So therefore, it's usually present in each generation, where recessive traits usually skip generations. There's a 50% risk of inheritance because you only need 1 copy of the allele to have the disorder. And 2 affected people can have an unaffected child, because now it's the a people that are normal, and the big A causes, the dominant allele causes the genetic defect. And of course, since it's autosomal, both males and females are affected with equal frequency. So here's an autosomal dominant pedigree. Notice that there's a lot more filled in symbols, right? Mom has it in generation 1, the first daughter, and the second son have it in generation 2. In generation 3, 4 people have it, both males and females. And then in generation 4, 2 males have it. So it's much more commonly expressed because it's dominant, and you only need one copy of the allele to show the phenotype. In this case, it's a genetic disorder. So some pedigrees here. So here mom has it, dad has it in the first generation, 3 of their 4 kids have it in the second generation, and then 1 kid has it in the third generation. If we add some phenotypes to this thing, we see that since they have a normal kid in generation 2, that tells us that dad has to be a heterozygote, and therefore the normal kid is little a. And defective people have to be big A, little a as well, because they get the big A from their dad, which gives them the disorder, and then the other allele has to be from the mom, and that's, of course, little a. So the last daughter there is big A, little a, and then her son that has it has to be big A, little a as well, because his father is normal, little a, little a. So examples of some diseases or disorders: Marfan syndrome is on the long arm of chromosome 15, and this causes the death of many athletes each year. The symptoms include a very tall, thin build with very long arms. Their arm span is greater than their height, and this makes these people good at playing basketball and volleyball. But they really shouldn't be athletes, because also weak aorta comes with that. And your aorta, remember, is the main blood vessel leaving your heart, carrying that arterial blood out. And when you're exercising, of course, your blood pressure shoots way up, and the aorta can break or burst. These people tend to be nearsighted as well. We think that Abe Lincoln had this. He had that phenotype, tall and thin. And then an Olympic volleyball player named Flo Hyman died from it, and I'll talk about that a little more later. Another disease that's much worse, though, is Huntington's disease. It's found in the short arm of chromosome 4, and it's a progressive degeneration of the nervous system. The worst thing about it is that it doesn't become expressed until late in life, usually after 30 years of age, when most people have already had kids and passed on the gene. And they usually die 5 to 15 years after the disease starts to make itself known. It's a degeneration of the nervous system where people have lots of involuntary shaking movements. Also, there's mental deterioration as well. So people with Marfan syndrome tend to, again, have very long arms, long fingers, and also kind of a caved-in chest with sort of a indentation between the two pectoralis muscles, I guess is the best way of saying it. So tall, thin people that tend to be stoop-shouldered and have long arm spans, like this fellow here, showing you a typical Marfan body type. Now, in 1960, a man sharing a common ancestor with Lincoln was diagnosed with Marfan syndrome. So now we know the allele was in his family, and he certainly had the phenotype. He was 6'5", which was ridiculously tall for the mid-19th century, and he had very long arms and hands and so on and so forth. So Lincoln probably suffered from it. Flo Hyman helped the U.S. Olympic volleyball team win a silver medal in the 1980s, I think. And she was she was 6'5" as well, very tall for a woman. And she was asked to be taken out of a volleyball game because she wasn't feeling well. And she sat down on the bench and slumped over dead. That was because her aorta burst. But they were able to save one of her brothers by going in and doing surgery. So she had a brother with it as well. So people with this shouldn't really be doing sports. And here's the Huntington gene on the short arm, towards the end of the short arm of chromosome 4. And it's one of these things that's a trinucleotide repeat. Okay, so just having too many CAG repeats in that gene dooms you to an early death and a degenerating nervous system. So people with the normal allele have from 11 to 34 of these CAG repeats in the gene, but people with the mutant allele have anywhere from 42 to 100 plus of these CAG repeats. And the more repeats you have, the earlier onset the disease is. Apparently there's a few cases of children coming down with it, so to speak, and they've got well over 100 CAG repeats. So a little bit of extra DNA in the wrong place, and you're basically screwed. This shows that Huntington's isn't the only disease, genetic disease that is caused by trinucleotide repeats, though. Several others, like Fragile X syndrome, we'll talk about that later. The normal range is 60 to 54 CGG repeats, but people with 50 to 1500 have that disorder, and there we can see Huntington's towards the kind of towards the bottom there. CAG 9 to 37 is normal, 37 to 121 is in the disease range. So moving on to X-linked recessive now. Here, males pass the trait to our daughters, so roughly half who become carriers, and the daughters then pass that trait on to half of their sons. So of course it's never transmitted father to son because sons get their X chromosome from their mothers, not their fathers. But it can be transmitted from maternal grandfather through mother to the grandson. And the phenotypic expression is much more common in males than in females because, of course, we males have only a single X chromosome. Whatever alleles are on that are expressed. We can't be heterozygous for X-linked traits like females can. So here is an X-linked, is a pedigree of an X-linked recessive disorder. We see in generation 1, mom there is a carrier. That means she's heterozygous. So their first daughter, person number 2 in generation 2 is a carrier, and then their son is affected with it. And then in generation 3, we see that the daughter in generation 2 has had a son that's affected, and the affected son in generation 2 has had a daughter who's a carrier. And then we can see a couple carrier females in generation 4 and a couple affected males in generation 4 as well. So, but you don't see any circles that are completely filled in, just squares. So that tells us that it's X-linked if it only shows up in males. Some pedigrees. So here, a carrier female and affected and a normal male have kids, and roughly half their sons will have the disorder, half will be normal, and all the girls will be normal, but half will be carriers, just depending on what X chromosome they inherit from their mother. So here are genotypes. So remember now, these X-linked traits, our alleles are superscripts in the X chromosome. So dad being normal is XAY, mama carrier is X big A, X little a, and their affected son is X little AY, getting, of course, the little a from his mom. The normal daughter, of course, is homozygous X big A, and then the carrier daughter has a genotype identical to her mother, the normal son a genotype identical to the father. So here, an affected male and a normal female, when they have kids, all the sons will be normal and all the daughters will be carriers. So looking at the genotypes of this pedigree, then we see that dad is X little AY, mom is X big A, and all the sons will be X big AY, because remember, the sons get their X from their mother. All the daughters will be carriers because they get one of their X's from their father, and all dad has to pass on is X little a's. In a rather uncommon instance where dad is affected and mom is a carrier, then we can, about half the sons will be affected, and some of the daughters can be affected. The daughters will either be carriers or be affected, but they can't have normal sons as well. So adding some genotypes to this, then we see mom is X big A, X little a. The affected males are X little AY, and then the normal male is X big AY, getting the X from his mom. And then the unusual instance when a female has it, she gets her X little a, one from dad and one from mom. Here's a pedigree where the mom is affected, the dad is normal. This time all the daughters will be carriers and all the sons will be affected. And so here's pedigree, genotypes added to that pedigree. So dad's X big AY, mom is X little a. So all the sons will be X little AY, all the daughters will be X big A, X little a. So here's a pedigree of red-green color blindness, which I suffer from. And it's fairly common in males. So generation 1, mom's carrier, dad's normal. They have a an affected son, followed by 3 normal kids, and then a carrier daughter. And then the carrier daughter has a carrier daughter and an affected son. The affected son has a carrier daughter, who then has an affected son in generation 4. And then the carrier daughter in generation 3 has an affected son in generation 4. So you see no filled in circles, just filled in squares. Only males have it, therefore it must be X-linked. So some examples: red-green color blindness. They say 8% of US males are affected with it. Hemophilia or bleeder's disease. It's present in 1 in 10,000 in males and 1 in 100,000 in females. It's quite prevalent in the royal families of Europe because, again, of consanguinity. Royalty are only supposed to marry other royalty. And so before long, we have first cousin matings. So here are some red-green color blindness tests. In the upper one on the right, I can see an orange 16. That's pretty much all I can see. Kind of a faint reddish pinkish 8 down in the lower left, and sort of a faint greenish 7 in the upper left. But otherwise, I don't see much in the way of numbers. I can tell traffic lights, however. So this shows hemophilia in the royal family of Europe. So Queen Victoria in generation 2 there was a carrier, and she had a son Leopold with it, a daughter Alice who was a carrier, and another daughter Beatrice who was a carrier. Alice had a daughter Irene and a daughter Alexandria who were both carriers, who took it to the Prussian royal family and the Russian royal family. The Russian royal family stops there in generation 5 because they were all executed as the Bolsheviks took over Russia and started the Soviet Union back around 1918. Let's see then. Beatrice had a daughter, Eugenie, who had it, married King Alfonso VIII of Spain, and they gave birth to 2 sons with it, and then it seemed to die out after that. And the same thing in the British royal family, it kind of died out as well. You can see no red circles, but plenty of red squares in this pedigree. So, before showing you this interesting little pedigree, there's really just 2 things you need to look for in this pedigree. Just 2. Is it present in every generation? If it is, then it's dominant. If it's not, it's recessive. Is it present in both sexes? If it's in both sexes, it's autosomal. If it's in males only, it is X-linked. So just look at those 2 things and you can determine the mode of inheritance. Okay, this is the Ptolemaic dynasty of Egypt that produced Cleopatra, the famous woman who had affairs and children with a couple of the Roman emperors. So we can see lots of consanguinity or inbreeding. We see, picking it up in about the 3rd generation, we see Bernanke and Ptolemy III, our first cousins. They have a son, Ptolemy IV, and a daughter, Arsinoe III. That brother and sister have Ptolemy V, who then marries Cleopatra of Syria, not the famous Cleopatra we're talking about. They have 4 kids: Ptolemy VI, the 7th, Cleopatra II, and Ptolemy IX. Ptolemy VII and his sister, Cleopatra II, have a daughter, Cleopatra III, who then marries her uncle, Ptolemy IX, to give birth to a daughter, Cleopatra Selene, Ptolemy X, and Cleopatra IV. And Ptolemy X, that playboy, has kids with both of his sisters, Cleopatra Selene and Cleopatra IV. With Cleopatra IV, he has Bernanke III. They have Bernanke III, who then mates with her uncle, Ptolemy the 11th. And then Cleopatra Selene and Ptolemy X have a daughter, Cleopatra IV, and a son, Ptolemy XIII, who have kids together, 2 of them. And then Ptolemy XIII has kids with another unidentified woman and has 2 more Ptolemies, Arsinoe IV, and then the famous Cleopatra is Cleopatra V. The reason she's red there is because she's the famous Cleopatra, not that she had any genetic disorder. Supposedly she was a beautiful woman. With all that inbreeding in her history, I'm a little skeptical about that. So anyway, this is kind of an interesting pedigree showing you lots of inbreeding. And in those days, people didn't obviously know anything about genetics, A, and B, thought royalty could only marry other royalty. So that sort of limits the gene pool, if you will. Now, moving on to chromosomal abnormalities. Chapter 4, we'll discuss aneuploidy and sex chromosomes. And then chapter 8, we'll discuss aneuploidy and autosomes and some chromosomal rearrangements that fairly commonly affect people. So with regards to aneuploidy, remember that's extra missing chromosomes. Down syndrome or trisomy 21 is a fairly common one. And so I may show you a karyotype 2n equals 47. That of course means they have 47 chromosomes. XX or XY means it can affect males or females, and plus 21 means that there's an extra chromosome 21. So if I gave you a pedigree like that, I would want you to be able to tell me what's wrong with that karyotype. If I gave you a karyotype like that, I'd want you to tell me what's wrong with that karyotype then. You wouldn't have to tell me a syndrome necessarily, just that there's an extra something or a missing something like that, okay? Turner syndrome is the only monosomy that humans can survive. That's monosomy for the X chromosome. So people with Turner syndrome are 2n equals 45. They have a single X chromosome and no second X or no Y chromosome. Klinefelter syndrome is when males have an extra X chromosome, so they're 2n equals 47, XY plus X. So they're really XXY. Patau syndrome, quite rare, trisomy 13. So people can be of either sex. 2n equals 17 plus 13 means an extra chromosome 13. Edwards syndrome, same thing there, trisomy 18. So it can affect either sexes. With Patau and Edwards, those children tend to only live for a few months after birth, maybe 3 to 4 months before dying. People with Down syndrome then can live to adulthood. So let's talk about some of these. Down syndrome occurs at about 1 in 700 live births, so less than 1%. Symptoms include anywhere from mild to severe mental retardation. Some of these people can finish high school. They have congenital heart defects. They tend to have a broad, short skull, 1% chance of getting leukemia, which is much higher than normal, hyperflexibility of joints, and then extra skin on the back of the neck. Even though this usually occurs from non-disjunction in women, especially older women, for reasons we've discussed before, 20 to 30% of the time, it occurs from non-disjunction in males, even though we're making fresh gametes all the time, we males are. So here's a G-banded karyotype of a male with Down syndrome. You can see the X and the Y chromosome there, and 3 number 21 chromosomes down at the bottom. So there's one extra of our smallest autosome, and people can be pretty messed up. This shows you about how the incidence of Down syndrome really increases with maternal age. So it's 1 in 2,000 in women who give birth in their late teens. By the time they're 30, it's 1 in 900. By the time they're 40, it's 1 in 100, so that's 1%. And then in the few instances where women who have had kids at age 50, it's about 1 in 12. They're approaching 10%. So the older the mother gets, the more likelihood there can be non-disjunction during meiosis. And remember, non-disjunction is spindle fiber failure. So here's the spindle apparatus. Again, the pale things in the middle are the chromosomes, and the darker things attached to them are the spindle fibers. And remember, that forms in women before you're born, so they're the same age as you are. So as you get over 35, the likelihood of those little protein fibers failing increases, as we saw in that graph a couple slides ago. Okay, there's a little boy with Down syndrome. Now, sometimes Down syndrome can run in families due to a translocation, a 1421 translocation. So here on the left, we've got the normal parent, and then on the right, we've got a translocation carrier. So they have one normal 14 chromosome, one normal 21 chromosome, and then they've got a 1421 translocation where the entire 21 is squished down on the end of 14. And so when they go through meiosis, they can produce gametes that have that 1421 translocation, normal gametes, gametes that have the translocated chromosome and 21, gametes with just chromosome 14, gametes with just chromosome 21, and then gametes with the translocation and 14. So basically, they have a very low chance, but it is possible for them to have normal kids. And this shows along the bottom there where it says zygotes, what happens when a normal gamete fertilizes these different gametes. So 2/3 of live births will either be translocation carrier or normal. 1/3 will have Down syndrome. And then the others will have either monosomy or trisomy for chromosomes 21 and 14. And those fetuses are miscarried or aborted before they are born. Now, Turner syndrome, again, the only monosomy we can survive, we humans, and that's monosomy for the X chromosome. So, of course, these people are females with underdeveloped ovaries, abnormal jaws sometimes, shield-like chests, and webbed necks. And they think these are due to single dosages of certain X-linked genes like it shows there. But there's 99% prenatal mortality in humans, but none in mice. So that's interesting. That could indicate there's different ways of dosage compensation in humans and mice. So only 1% of the time do Turner syndrome fetuses make it to birth, but they can survive to adulthood apparently. So here's somebody with Turner syndrome, only 45 chromosomes, just a single X chromosome. This shows a patient showing you they have skin folds on either side of the neck and a lower than normal hairline. These people are female, but they never undergo puberty. They're sterile, never develop breasts or widened hips or anything like that. They're like prepubescent girls their whole life. Now, Klinefelter syndrome is where a male gets an extra X chromosome. So we see down here, all the autosomes are normal, and then down on the lower right, there's 2 X's and a Y. So of course, the Y chromosome will make that person a male, but they may not be phenotypically a normal male having that extra X. Like we see here with this guy, he has smaller than normal genitalia and a pretty narrow set of shoulders. However, some men with Klinefelters can be totally normal looking anyway. Now let's look at the rearrangements of individual chromosomes instead of aneuploidy. So Cre-du-Chat syndrome, that's 2n equals 46, XX or XY, 5p minus. So normal number of chromosomes, male or female, 5 means chromosome 5, p means the short arm of chromosome 5, and minus means a deletion. If there's a duplication, we'd say plus. The long arm of a chromosome is called the Q arm, the short arm is always the P arm. Okay? Fragile X syndrome, this is when the tip of the X chromosome breaks off or becomes unraveled. So we'll discuss these two. And this is a table from an older version of your textbook, but from table or chapter 9, excuse me. And the more common syndromes actually have names like Cre-du-Chat, Wolff-Hirschhorn, Prader-Willi. The other ones where the syndromes don't have a name means they're really quite rare, but many affect the phenotype of the head and face and also cause severe mental retardation. So in Cre-du-Chat syndrome, symptoms include microcephaly, that is a smaller than normal head, severe mental retardation, congenital heart disease, and a deformed larynx in about half of the kids that have it, causing them to sound like a cat when crying, and that's what Cre-du-Chat is. It's French for cry of the cat, so it's cry of the cat syndrome. So here's a little boy that's got it, showing you microcephaly or a smaller than normal head. And then here's a karyotype. So it looks almost completely normal, doesn't it, for a male with Cre-du-Chat syndrome? But if you look at the right chromosome 5 where that arrow is, you can see a little bit of that short arm is missing. Even though the other chromosome 5 is totally normal, the kid is severely messed up. So you need both complete normal chromosome 5s to be normal. Now in Fragile X syndrome, the frequency of this, of course, is more common in males than in females because it's X-linked, so it's 1 in 1250 males or 1 in 2000 females. Symptoms, it's the most common form of inherited mental retardation. There are altered speech patterns where the people speak very slowly with pauses in their speech in sort of unusual, inappropriate places, and the males with this have larger than normal testes. The fragile site, the reason this happens is because there is a fragile site towards the end of chromosome, the X chromosome. Fragile sites are places along chromosomes where they tend to break more than usual. And there are, and this is due to an increased number of trinucleotide repeats, in this case CGG, not CAG as in Huntington's, but CGG in this case. So this shows a normal X chromosome on the left and one with Fragile X on the right. Here is a female with Fragile X at the top, a male with it on the top right. And interestingly, a pedigree there where there's a female who's a carrier, and she has a son who's a carrier, then 3 normal sons, and then a son with the trait with Fragile X. The carrier son then mates with a normal woman, but they have 4 sons, and 3 of those 4 sons have Fragile X syndrome. And this shows you the gene. FMR1 is the name of the gene. That stands for Fragile Mental Retardation 1 gene. And what causes this is expanded repeat. So the DNA repeats there are shown to the left of the gene with that bluish area with the little vertical lines in it. So expanded repeats, that's more than normal number of CGG repeats, causes the gene to be expressed and the tip of the X to break off, where people are then have inherited mental retardation. So this shows you fragile sites on human chromosome 16. This doesn't cause Fragile X, but it illustrates fragile sites. So we can see that in both of these 16s there, they break in the proximal place and not in the distal place, at least in that one sister chromatin. You can see that on the top as well. So fragile sites are places where chromosomes tend to break more. They're not just in humans, they're in all kinds of creatures. So with that, I'd like you to do problems 26, 27, and 29 in chapter 6. These are pedigree problems, and I'll email out a due date. And that's all I have for human genetics.